What is up, guys? So I have an anatomy quiz today in about an hour and a half. Yeah. So I'm going to be going over some of the stuff I discussed in the previous video and some of the new material we learned to try and prepare for this quiz. So I'm going to be going over some of the figures discussed in the textbook and the tables, and a bit of some of the lecture readings and notes we had. Just put a bit of lighting over here so I can shine my book. Okay. First one, first figure we talked about was this one. Of course, as we discussed in last video, the sensory pathway. So first we have stimulus that has to come in, then there's a depolarization, produces an action potential. Propagation, then it's that information is processed in the central nervous system. If that information is voluntary or the sensation, then it goes to a motor pathway for a voluntary response, or in other words, goes to an effector. If it's involuntary, it goes to an immediate response and acts as a reflex type of response, or an immediate response. It's not really processed at the cerebral cortex, it's just related to a synapse for sensory information. So the circuit begins. The organ receiving the stimulus is a receptor. At some point, the stimulus assists the receptor. The receptor is divided into three areas. Organization of the nervous and organ system. So there's special sensory receptors, visceral receptors, and somatic skin receptors that come from muscles, joints, and the skin. The stimulus arises at the receptor and the muscles reach a threshold, so the stimulus goes through a chemical gate. An action potential which is propagated the central nervous system for where there is processing. If it reaches the cerebral cortex, where it becomes a perception. A sensation is a stimulus that is received that makes it to the central nervous system. Its effects may be similar to a reflex. To be aware of it, some of the conscious and subconscious parts of science, perception versus sensation. So intensity is when it comes in as a stimulus, but how it is transferred through a circuit is different, since we said an action potential can be strong or weak. It's based on how many neurons are receiving the information, or it can be a rapid firing of neurons reaching threshold very often, or a lot of neurons honed to the same receptor. Afferent division is more on the loudness or the strength of the signal, how intense it is. Intense stimulus versus weak stimulus, based on how rapidly neurons are being fired down. General senses, as opposed to special senses, are senses laid out throughout your body, has a field sensation area on one spindle fiber and muscle relaying information about a certain group of muscles. Sensory, and then the next day I think this is the quiz day, we talked about the same figure again on the overview events of the sensory motor pathways. Or a bit of recap: involuntary and voluntarily response. If it is not, then it's visceral. Three types of senses include general, somatic, visceral, and special senses. They bring out an action potential that gets sent to the central nervous system for a reflexive or voluntary response. Okay, and a bit of these, a figure of these receptors and their receptive fields. You can see that there's some axons and dendrites down here receiving signal, some sending it to these axons, the dendrites. So receptors here receiving a signal. Each receptor cell monitors a specific area known as the receptive field. So how do we classify senses? As general, having somatic origin. Covers the senses of touch, pressure, pain, temperature, vibration, proprioception, position of muscles, extension of your joints, sensory organs, Golgi apparatus. Joints have senses that sense how much they have been moved. Some general senses include touch, pressure, pain, and vibration. Received by receptors, so the different types of receptor types include mechanoreceptors. These are touch receptors, pressure, and baroreceptors. Dermoreceptors sense temperature, so general ones are all over the skin, below the epidermis. They're in two different places in our body, the skin and the hypothalamus. They are sensed by the hypothalamus, so the thermoreceptors in the hypothalamus ensuring proper temperature. Proprioception allows for a sense of body position. Nociceptors, sense of pain. Photoreceptors, retina of the eyes. Chemoreceptors, sense of smell and taste. Mechano is more on equilibrium of the inner ear and hearing. Noci is pain. Photo is vision. Chemo is smell and taste. So the five main types of receptors include chemoreceptors, mechanoreceptors, nociceptors, photoreceptors, chemoreceptors, mechano, nociceptors, photoreceptors. What was the last one? Thermoreceptors. Thermoreceptors are more on temperature. Can't forget those. With the sixth one that can also be included in this category is proprioceptors, as part of the five main types, even though it can be a subtype as for mechanoreceptors. Okay. So the next figure we're going to be talking about is this one. So in this figure, we discussed this in the last video, but you can see the free nerve endings, root hair plexus, the different types of parts of the skin or tactile receptors on the skin, dendrites, the dermis, sensory nerve fibers, and the different types of corpuscles. Right there. Yeah, I highlight a lot of my books. So chemoreceptors are near the center of medulla oblongata. They sense oxygen, carbon dioxide, and regulate pH. Now we're moving on to figures from chapter 17. So now we're going into olfactory organs. So a bit of the epithelium there. You can see the olfactory nerve right there. Olfactory bulbs, tract, and the central nervous system. Olfactory receptor is a modified neuron with multiple cilia dendrites. So you can see the knob and the dendrites. So nerve fibers. Okay, alright. So conic versus phasic receptors. When information comes in at greater strength, the interval of the impulse is sped up. When it comes in at a lesser strength, the interval is slowed down, but they are sensing constantly. Conic receptors receive information at regular intervals. Phasic receptors sense information at periodic intervals. If you have fast adapting senses, it means you're not aware anymore of incoming sensations, touch, pressure, and smell. If you have slow adapting, it means your senses, you're still aware. So here's a bit of a better picture of your senses. So olfaction, smell, and gustation, taste. So you can see olfactory receptors are dendrites, specialized neurons. Olfactory receptors, dendrites, this is a specialized neuron. Picks up information, goes down axons leading to the central nervous system for processing. When the dissolved chemicals contact the dendritic process, there's a depolarization called a generator potential. Yep. And this graph shows the actual potential produced by a generator potential. Olfaction, gustation, smell sensitive gives information about our environment. Gustation, taste. Because there's a synapse involved, there's a slight synaptic delay. So here's the synapse. between receptor cell. Because taste has different from smell. Taste uses receptor cells. And chemical transmitters are released at this synapse, creating a generative potential. Yep. Yep. Stimulus is picked up by a neuron, receptor cell. Stimulus hits a sensitive cell, which then speaks to its neuron. We don't need to know the G protein, cyclic AMP, and stuff like that. When stimulus arrives at a receptor cell, then the input gradient reaches to threshold. Receptor potential goes up to a graded potential. Graded potential summates to make an action potential. At the initial stimulus level, the graded potential is called a receptor potential. Receptor potential goes on to make an action potential. Okay. Okay, and then going back to that figure about the olfactory one with the nose and the senses. Olfaction sense, it's least evolved sense. Ability to sense chemicals in liquid as a molecule and odorants, these extra long dendrites. The odorant is picked up by olfactory dendrites. Neurons have developed extra long dendrites to receive odorants. Odorants are chemical molecules that are in the sense of smell. Need a level of moisture in order to smell the odorants. Okay, I need to go through this a bit faster because I need to get to the new material already. So olfaction, smelling, is a sense that can go straight to your hypothalamus. It's a direct one. So now I'm going on to taste. So for taste, we talk about this figure. So gustatory receptors, taste buds, umami, papillae, taste papilla, that type of stuff. Taste papilla, little bumps that we thought were taste buds. So different shape, they're based on that figure I just showed you guys. There are different shapes of papilla. Spongiform, filiform. Spongiform is like fungi, filiform is like a leaf. Papilla have a crack in each side where saliva is kept and dissolved in saliva are the tastent molecules. They sit in these cracks until taste buds take them in across their membrane for the formation of an action potential. Okay, onto the ear. The inner ear is responsible for equilibrium of rotational position and acceleration up, down, forward balance. So there's 3 semicircular canals. There's a middle ear. The middle ear is involved in detection of sound in the cochlea. Mechanical bending, moving, other types of equilibrium takes place in the vestibule. There's 3 special sense structures: the cochlea, the vestibule, and the central canal for rotation and body position. Hearing relies on all 3 of these. Semicircular canals are filled with ducts. The fluid inside the ducts are called lymph. Canals are bony structures and ducts are socket tubes filled with lymph fluid. There's perilymph, endolymph, not just water. Lymph fluids also water. Water is allowing them to work right. Most of these lymphs, including, are completely thickened, not just like water. Essentially, movement of fluids at 3 canals trigger ampullae. Inside of each is a drum structure. It is a thickened form for lymph fluid. Drum-shaped structure. Ampullae of the thickened form of lymph fluid. Endolymph fluid gets thickened into a gel. The gel bends into hair cells that are bent to a certain degree. The cupula will pull on them. This tells you your position in space, tilt and bending gel and bending hairs. And then the day of my last week's quiz, we got a bunch of notes. So I'm just gonna go over them with you guys real quick. Okay. So on the quiz, we got quizzed on the 5 main types of receptors. Okay. So the 5 main types: nociceptors, thermoreceptors, chemoreceptors, mechanoreceptors, photoreceptors, and also kind of proprioceptors. The second one, so what is transduction? So like translating something. Duction is electrical signal. Modalities are stimulus such as sound waves, vision, and light waves that we receive. The body needs to translate these stimulus into a specific language of electrochemical and action from a neuron. In other words, an action potential. It's the language of electrical conduction as an action potential. 2 special chemical senses include smell, olfaction, and taste, gustation. So back to that figure that we discussed a while ago, showing the tongue and other different types of taste. Humans have basic taste abilities. So we have 5 senses: salty, sweet, bitter, sour, umami, the fifth one. So the book talks about where different kinds of taste buds are located, the different types of papilla. You don't need to know the different taste sense and papilla. So umami. Umami means savory. It's identified scientifically as a taste of protein. There's some adaptive significance to this, so taste changes by gender, age, your physiological state. Why do we have taste? Has to do with survival value. Taste in infants change a lot for the first few years. Infants have heightened ability to taste bitter foods and things. Infants have an amazing olfactory ability. So they have amazing ability to smell things. Olfactory. So they have amazing taste and smell. We have an affinity for sweet taste because of glucose. Okay, next figure. This one's far ahead. Next figure is on the anatomy of the ear. So you can see here, you have an external ear, middle ear, and an internal ear. So external ear is the thing we usually see on our ears right there. And going inside, we get to the middle ear, where we have our eardrum or tympanic membrane. That's our eardrum right there. And then into the internal ear, you can see some of the important things, such as semicircular canals, those three things, and the snail or the cochlea. And the some nerves here: facial nerve, vestibulocochlear nerve for balance. Cochlea is also for balance and equilibrium. Then you can see the vestibule right there. The round window. Auditory tube leading to your nasal pharynx. So some liquids that go into the ear can reach your nasal pharynx if it actually gets past that tympanic membrane or eardrum. So equilibrium. Equilibrium takes place in one small portion, the vestibule and the three semicircular canals. Comes in two types and in three senses: semicircular canals that detect tilt and position of the head. The inner ear begins where the stirrup meets the oval window. The oval window. Huh. When the oval window vibrates, it is innervated by the liquid gets vibrated. The duct containing lymph fluid inside. The soft duct is endolymph, and it's innervated by the stirrup. Okay. Sensitive hair cells. Sensitive hair cells are sensitive long hairs containing cilia bent by a thick endolymph so that it doesn't slosh, it just moves in a certain direction. Next one is a figure of the internal ear. So if there's a cross section, you can see the different layers. There's the endolymph that we were talking about, the perilymph around it, and the bony labyrinth on the outside. So there's a semicircular ducts. Looks like a ribbon. Semicircular canals. There's the utricle and sacculae. Sacculae is closer to the snail. Utricle is closer to the semicircular canals. Vestibular duct, the tympanic duct, the cochlea of the snail, the maculae, right there, the purple stuff, and the cristae of ampullae is right there on the closer to the utricle. The maculae is closer to the sacculae. Yep. So that's a better view of the internal ear. So the inner ear is a hard duct with soft tube, within a soft tube. Endolymph takes a thickened liquid form in the vestibule. Thickened version of water, so that hair is not just flapping. Sensitive hair cells are receptive cells. Helps your brain be consciously aware of potentials in space. Okay, we're going to be going through a bunch of figures now. So now, a bit of the crista ampullae, different directions of duct rotation. This describes the endolymph movement along the length of the duct, moving the cupula. And the semicircular duct is stimulating hair cells. You can see it starts moving, goes here, and it's like a pendulum, back and forth, back and forth. And that movement is based on endolymph moving those cupula containing the sensitive hair cells. And that tells you position. Ampullae, crista ampullaris, so the utricle is close to a U, which looks like a semicircular canal. Sacculae is close to the snail. Okay, this figure is of, now we're talking about this figure. So representative of a hair cell, so inside the cupula. There's bending stereocilia toward the kinocilum. So there's stereocilia on the top. There's a hair cell, some sensory nerve endings, a supporting cell. Mechanism of utricle and sacculae have sensitive hair cells leading to neurons, which join to axons to form a nerve. So that yellow part was leading to a neuron, and which will later reach an axon. To form a nerve, vestibulocochlear nerve specifically. Next figure, a closer view of the sacculae and the utricle. So we move our head up. Our hair cells bend like that compared to being straight like that because of gravity. So shifting tells our head the position. We have a closer look at the maculae. Maculae has a lot of otoliths. They look like little stones. And you can see there's also some hair cells here. And nerve fibers. Hair cells join together with axons forming vestibulocochlear. Otoliths is a thickened membrane, so little stones. Lith stands for stones. Otoliths, so when you move otoliths are little stones that move the stones and gels to trigger a movement. An example would be in the elevator. Back to the last figure with the hair cell. There's stereocilia and to help describe this mechanism. Next on to this figure, more on the nervous system part. You can see the vestibular ganglion, the vestibule, semicircular canals, and you can see a vestibular branch connecting to the pons. The terms of nerves being innervated here. Vestibular nucleus, this one's linked to the cerebellum. So lots of information process of the positions of things. Okay, nerve vestibule comes to pons and up to the cerebral cortex. Information is used by the cerebellum to adjust to cerebral output. The vestibule joins with the cochlear branch, forming the vestibular cochlear nerve number 8. The inner ear is more sterile. And the inner ear is more sterile, have microbes that don't belong there. Man, there's a lot of pictures here. So, here's the next figure with the auditory ossicles. You can see the malleus, the incus, and the stapes. The stapes connects to the oval window that we were talking about before. There's the tympanic membrane. There's the round window, so that starts out the inner ear. So the oval window starts at the inner ear. So stapes is a type of auditory ossicle. Okay. It's not meant to be fluid-filled, but air-filled. 3 bones, the malleus, incus, and stapes. Oh, shoot. Show nerve transfer and amplify vibrations from one side. Transferring vibrations to the oval window where vibrations are sent to the inner ear. Molecules of air pulsed. Next figure. Holy cow. There's so many figures. The nature of sound, so types of amplitude. Amplitude is a height, wavelength, sound intensity over time. Sound waves are transmitted to the inner ear as they head through external and internal ear canal. Wavelength is the distance between two peaks. Frequency is how many times you reach a peak during a period of time. Amplitude is the height of the wavelength. So if you have a long wavelength, it means you have shorter frequency. If you have a high wavelength, then you have high frequency. Or short wavelength would mean higher frequency. Actually, I think I put that in wrong. So here's a cross-section of the cochlea here. You can see the different bones. It's vestibular membrane, basilar membrane, cochlea. Yeah. Some nerves here, tectorial membrane, vestibular membrane, basilar membrane. There's the front oval window, the round window below. So this is all coming from the snail. Yeah. Between spaces is a basilar membrane that is the basilar membrane is the mechanism of transduction. So like we discussed before, changing that stimulus into an action potential, translating it. The organ, so there's some sensitive hair cells. Organ of Corti, within each little gray patch. We have hair cells. So now in this figure, cochlear duct, here's a bit of a microscopic view. You can see some hair cells, some spiral gang organ. Inner hair cells, there's the tectorial membrane and the basilar membrane below it, outer hair cells, inner hair cells. And some nerve fibers right there. So hair cells sound waves come into a liquid and bends a membrane, working its way around the round window, pushing against connective tissue cells, bending basilar membrane. Distinctions help the brain distinguish different sounds and their pitch. So wave is now in liquid, pushing on the basilar membrane, pushing on hair cells, connected to the bending every time it hits the basilar membrane. Cautions hair cells to bend at a certain degree. Next figure is the frequency discrimination. So a bit of the actual basilar membrane in the cochlea bending based on the degree of the stimulus. So there's the stapes and the round window. Stapes and round window are all affected by the basilar membrane's movement. And that information is processed at the central nervous system based on how this distortion goes at different frequencies. So the basilar membrane bends hair cells, telling the brain through these neurons that it is a loud sound. The amplitude of the wave bends the cells more. We have an auditory cortex that reads some as a soft sound that is not enough to reach the threshold that some humans cannot hear. Loud sound causes basilar membrane to bend a lot, causing hair cells to bend, move. Amplitude registers in the brain as loudness or volume. Next is on the pathways side of things. So stimulation of hair cells, bending, goes down the vestibular branch, carrying the sound information to the vestibulocochlear nerve. Information is sent to each cochlea nucleus. Reticular formation. Inferior colliculi directs a variety of unconscious motor responses to sound. Goes up to the thalamus, the relay center. Sending acoustic information goes to the mediogenicular nucleus. Projection fibers then deliver information to specific locations on the auditory cortex of the temporal lobe. So this is the cortex up there. Okay. So the pitch story, based on frequency of waves, based on how many ups there are in a unit of time. High frequency, short frequency waves hit the membrane so rapidly that it exhausts its energy quickly, doing a lot of lowering and dissipates energy early in the cochlea. If the brain gets a high frequency, it is transformed into a high pitch. High frequency sound means high pitch. Low frequency sound works way up the snail, not dissipating. In hair cells translated as low sound or low pitch. Heads into the brain, trigger bends in hair cells translate as low frequency sounds translate as low pitch, heads into the brain at the pons, out the... So there's a bend in the hair cells as low frequency sound is translated as low pitch, up cochlear nerve, vestibular cochlear nerve, heading into the brain at the pons, out the thalamus and straight to the cortex. To areas of association with hearing. Association areas. Starts out at primary sensory gyrus, doesn't synapse until it reaches the occipital lobe. Thalamus, the third order neuron, through primary sensory cortex, the ventral gyrus. Associate the sensory organs of gyrus maybe to cranium to make a decision. Everything goes up the gyrus and goes to association areas in the cortex. The primary central gyrus, everything synapses at this association area. And just like that, that's the equilibrium story of the ear. That's a lot of information. Now we're moving on to the eye. Thank goodness. Holy cow, that was a lot of information on the ear. So, what about the eye? Okay. Dang, there's not much time. Gotta do this fast. So here's a bit of a view. There's lacrimal gland. There's a conjunctiva there. Some obliques on the bottom. Okay. So we have a pupil, the region with no iris, a hole heading straight into your eye, fortunately covered by a glassy piece called a cornea. Cornea is a transparent portion of the sclera, the bigger eyeball. If followed, the sclera goes around the eye and becomes white. Accessory structures, we have eyelids or lids that have a lacrimal gland, or lateral / superior to the eyeball. Ducted accessory gland ducts fluid into surface of cornea from lateral / superior to medial of the eye. Comes from a tear gland to clean and lubricate bacteria. Side and nourish the cornea. Purpose of tears. So tears contain lysozymes, an enzyme that breaks apart the bacterial walls and has lots of nutrients as well. Parasympathetic division of the nervous system gets involved with the eye by stimulating tear glands based on resistance, tears because irritation or sad. So a bit of the pupillary muscle in the eye, pupillary dilator muscles. Contraction of these muscles enlarge the pupil. So constrictor muscles and dilator radial muscles. Constrictor muscles are concentric circle, so circular muscles. So to contract, constrict, circular. They use parasympathetic division. Dilation uses sympathetic. Very much opposite. Muscles in the iris have two layers. The circular is the inner, the radial is the outer. Constrictor layer is contracted by the parasympathetic nervous system. Sympathetic nervous system dilates pupil and contracted. Sympathetic nervous system radiates smooth muscles, dilating pupils for contraction. Next figure, saying -5A, is a fornix, conjunctiva, serratia, the cornea, pupil, the lens, iris, the retina, and then the choroid layer is below the retina, and then the sclera is on the outside. Fovea is right there, optic nerve. Yeah. Lids, lacrimal glands, iris, pupil, cornea. The iris in the back of its iris is regulators of color, backed by a narrow pigmented layer. Yeah. Its function is to prevent stray rays of light or photons from going where you don't want them to go. The eye is designed to project photons away. You should not be bouncing around on you, or you will get lots of flashes of input. Images. Ensures light is coming through the pupil to a specific type of pupil to a specific part of the eye. Conjunctiva, pale pink layer like a gasket between accessory structures of a skin and eyeball itself. Example, it keeps your contact lens from going back to the brain. When tear suspect lens are blocked, there will be inflammation. Lacrimal gland and tears usually protect the eye effectively. You have salivary and lacrimal glands, which will require a good dose of liquid all the time. An example is dried of liquid. So that's why we should keep ourselves hydrated. And now to the latest lecture. Okay, we can't stop now. Yeah, keep going. We just caught up. So we just recapped on the quiz in the beginning. 5 types of receptors and the transduction. What is transduction? It's the conversion of stimulus to an action potential. So I got these are the results from that last quiz. Extra credit points. Yep. Okay. Let's jump into this array. So we're still on the same figure. Accessory structures. Lacrimal gland, lids, conjunctiva, the gasket or lid, your eyeball and sclera. Sclera is made up of dense fibrous connective tissue. It's the bulge in front of the cornea. Bulge in front of the cornea, it is the outlet in the back of the optic nerve. So we have 3 tunics. We have a fibrous tunic, which is the outer one, made up of the cornea and the sclera. Fibrous cornea. Sclera. Continuous structure where this fibrous connective tissue becomes transparent and bulged at some point in the sphere. Our FVN. Okay. Our eyes have different shapes, so it can be elongated, bulged. That affects the accommodation story. The cornea is bounded by a conjunctiva in a thin membrane over the surface. The cornea and sclera are made up of a lot of collagen. Conjunctiva contains tiny blood vessels, bloodshot eyes, capillaries or blood. That means capillaries or vessels are broken and repair is needed quickly. Repairs quickly. The bulb is covered with collagen. The purpose of this dense tough ball is to protect the delicate things inside. Allows for muscles to attach to it also. The vascular tunic, or the middle layer, is the tunic inside the ball. It's vascularized, so of course there's lots of vessels in there. Also known as the uvea. Tunic is the tunic inside the bulb. That's what that means, uvea. It has the anterior portion has ciliary body. Their serrated edges darken regions. Once sliced through, you can see it with little threads at the edge called ciliary processes that suspend the lens on the ciliary body. Suspensory ligament. So ciliary processes are where lens is attached by tiny threads. Capillaries capillaries secrete aqueous humor that bathes the front of the eye. They'll be distracted. Capillaries secrete aqueous humor that bathes the front of the eye. Capillaries secrete plasma that is filtered. Comes out to be aqueous humor that is nourishing and is bactericidal. The anterior cavity is divided into two chambers. So we have an anterior chamber that is in the front of the iris, and a posterior chamber of the anterior cavity is in the front of the lens. We have an anterior chamber that is in front of the iris from the lens. So these are still in the vascular chain. Aqueous humor exists in the front of the lens, divided into two pockets, the anterior and posterior. The iris is a dividing peninsula dividing the same liquid aqueous humor dividing this extension of ciliary body that determines how much light hits each lens, determines how much light comes in also. Controlled by circulating and radiating muscles, controlled by... So iris is responsible for determining how much light comes in into the lens, controlled by circulating and radiating muscles, controlled by either sympathetic and parasympathetic. So sympathetic division for the iris is radiating muscles, contract the eyes, opening the pupil. Parasympathetic is circular muscles that close the space, constricting the eye. Sympathetic is more on dilation here, using radiating muscles. Parasympathetic uses circular muscles more on contracting or constricting that pupil. The iris is the nice colored part of the eye, that dark pigment, light absorbing cells. In addition is the ciliary body. Iris is human pupil. There is lens, keeps stray light from bouncing all over in the retina. Lens is squashed egg shaped, attached by suspensory ligament. Very distinctly dilated pupils means nerve damage. Next figure shows the different layers. Fibrous layer, cornea, sclera. Vascular layer, you can see the iris, ciliary body, the choroid, anterior cavity, posterior cavity, neural. Okay, so posterior of the eyeball. So the posterior cavity of the eyeball has a choroid layer. Darkened layer of cells, part of the thickened vascular layer. Um, the choroid is the intravascular where blood supply is brought in and nourishes the retina of the eye. It's full of melanocytes that send fingers into the dermal layer, protects cell nuclei from sunlight. Choroid layer absorbs stray light. Animals with night vision reflects choroid to the retina. Third one is the third tunic is the neural tunic. It's the innermost tunic about the nervous system, not very vascular system. Retina is the orange layer. It's a very active layer of light sensitive cells. Transduction takes place in these cells. Several layers form the retina because they are forming converging circuitry. Lots of receptor cells are speaking to fewer neurons and eventually combining information and sending them out to the optic nerve, allowing you to have extreme sensitivity to lots of rods and cones that feed few ganglion cells. So it goes to the ganglion cells, then to the optic nerve. Next, we talk about this figure. It's the organization of the retina. So it's a bit of a slice of a retina there. You can see the rods and cones, the choroid, the glands, some of the ganglion cells. So when light comes in, it goes to the ganglion cells, then it reaches the rods and cones, and then that information goes to the optic nerve. So there's a slice of the retina. We have sensitive cells, a choroid layer in the back, and a skinny layer of a retina. Starts in the back with the skinny layer. Rods and cones look different in shape. It is made up of a layer that reaches pigments embedded into a background of pigment and epithelium. Okay. So next figure is this figure, so you can see the pigment epithelium. So light comes in, reaches bipolar cells. Each photoreceptor synapses with a bipolar cell. Then it reaches rods and cones. Then it reaches this, and then it reaches pigmented epithelium, which absorbs photons that are absorbed by visual pigments. It is also called phagocytes. So there's rod-like receptors and cone-like receptors that are embedded into the backbone of the pigmented epithelium that ensures that light is reabsorbed into the pigmented layer. So it's all about getting that light reabsorbed into the pigmented layer. Rods are sensitive to shades of gray, black, and white, no color, much more sensitive to dim light. Example, we walk around in moonlight. Yeah. So you want a good color vision in dim light, picking up light at a lower level. Information gets sent to a layer of neurons inside them called a neuron. Starts at a junction between bipolar cells. So we go from bipolar cells to ganglion cells. Bipolar cells to ganglion cells to rods and cones to pigmented epithelium. Cones pick up wavelength of different colors but require more light. Sensitive rods and cones send information to layers of nerve. So ganglion cells to the back of the eyeball, basically. Departure of the optic nerve, not directly behind pupil, off center. Not directly behind pupil, off center, has to carry information to your brain. So after it leaves the optic nerve, we're going straight to the brain. Entire retina is capable of receiving light and get a focus image from part of your retina directly behind the pupil at focusing points called the fovea. The macula is a spot at the center containing a middle point called the fovea. The fovea. The blind spot is where the brain tries to convince itself that it doesn't have a blind spot because there's no rods and cones and no vision at all. So image formation. Yeah. Any image you look at will be produced. So we're looking at this picture of an image formation of looking at a electric power stick thing. I forgot what that's called. Any image you look at will be projected, will have some height, depth, and dimensions to your retina as an image upside down. It can be backwards. Yeah. If it's focused on the upper portion of the retina. Light is projected to your lens and projected back to your retina in a straight line so you can see something upside down. You can only focus on a pole that your eye is focusing on. You can't focus on the whole pole. In order to focus on the pole, you use your muscles to move your eyeball so that it's turning directly behind the pupil, at the bottom of the pupil. In order to focus on anything, the eyeball needs to be turned so that it can be projected into the fovea to be focused on. On the retina, it is backwards and upside down. It is sent to the brain so it can be seen right side up. So here's some factors affecting focal distance. Light rays from a source are refracted when they reach the lens of the eye. When they reach the lens, so it's refracted. So it reaches focal point. So what is happening with light? Light is reflecting off itself, bending light and projecting it onto the fovea. It's projecting onto the fovea. Lens bends light and is expressed at the fovea. Light goes from air to a dense fibrous to gelatinous liquid. When looking at something far away, light is reflected off it so that light has to bend at a certain degree, bending at an angle. Lens has to do something for the focal point to be at the fovea of the eyeball. Making sure light is bent, hits your fovea. The lens is rounded so the fovea bends light more. A curved object will. Yeah. So for close vision, ciliary muscles contracted. When those ciliary muscles are contracted, the lens is rounded. Focal point is on the fovea. When the muscles are relaxed, the lens is flattened, which is weird. Okay. Curved object will bend light and flat object is send light straight through. Close vision, so accommodation. Ciliary muscles are contracted, lens is rounded. As ciliary muscles contract, they round. The sphere gets light focal point to meet at the fovea. Focal point has to meet at the fovea, tell lens to bend so that they can meet up at your fovea. For 20/20 vision, so that's for close vision. For distant vision, relaxing the eyes, ciliary muscles relaxed, lens is flattened. Focal point meeting at the fovea makes lens to bend light enough so that the rays bend and meet your fovea. All about distance. There's two problems when you get older. Your lens do not flex well enough. Lens or your ciliary muscles in particular gets harder, making it hard to round it and to focus. Muscles cannot be rounded enough to meet focal point or fovea. Accommodation, so the muscles, the more reasons for problems would be the muscle gets weaker. Eyeballs cannot be perfect spheres. Lens is not curving enough. Eyeball can also not be too long or too short to bring light to the right spot. Using another lens, we use glasses sometimes to help rays get in a straighter angle. Artificial accommodation with glasses or contact lenses. Back to this figure of the pigmented fluid outer segment and inner segment. So we have photoreceptors, bioreceptor cells. Rods are sensitive to black, white, and shades of gray. at subcellular level, cones are sensitive to different wavelengths of light, different colors of light. This figure again with sodium, more on the chemical side of things. Sodium entry through gated channels produces a dark current. Opsin activation, and a bit of rhodopsin here. Don't need to know visual light spectrum. Cones are sensitive to wavelengths of light. Cones are sensitive to different wavelengths of light. Several different groups of cones always working together. If you have some cones working properly, then you are colorblind. Photoreception involves a series of enzymes induced that take place inside the sensitive cells. Have pigments that absorb different wavelengths of light. Of specific absorbance, absorbing certain colors, reflecting the rest. Retinal is a carotenoid. Retinal is a carotenoid association between vitamin A component and pigment. We also have photopigments. Photopigment absorbs certain colors and reflects the rest. The pigment is called the retinal. Example would be eating carrots improves retinal pigments. Retinal is vitamin A pigmented component. Retinal plus opsin produces rhodopsin. Opsin is a glycoprotein. Glyco because carbohydrate, protein because it's a protein. Carbohydrate protein. Rhodopsin gets split into two components, retinal and opsin, when hit by light. This is called bleaching of photopigments. Photopigments get split apart until pigments are not able to receive light, but they still. So when light hits it splits apart. But they're still able to. When light hits the retina, photopigments split apart, and retina can't receive light. But they rhodopsin are constantly regenerate or rebuild itself quickly after damage. Rhodopsin rebuilds itself. Rods are more sensitive to levels of light and take longer to put themselves together. A bit about this. Back on that figure with the sodium. Process of photoreception is a process of destruction. In the dark, there is a dark current operating, so the number of neurons. Sodium gates are open process of neuroglia. Backwards, only a little bit of sodium comes into the neurons. It's very backwards, this process, because only a little bit of sodium comes into the neuron. Resting potential is at -40. Active state closes sodium gates, dropping action potential to below -70. Process of receiving light, taking light in and breaking it into pieces. When light hits photoreceptors, light closes sodium gates, causing resting potential to return to -70. In the eye, this neuroactivity is backwards, taking light in and breaking it apart to individual pieces. Difference between rods and cones is the ability to regenerate, which is more sensitive. Rods are more sensitive but takes longer to put together. Cones are faster but are put together more rapidly. Breaking apart and regenerating of many rods and cones. Rods take longer to regenerate. It takes a while before you can see a new image because it's black and white. You need to see color more. Cones regenerate fast, regenerate in a few minutes. Visual issues relative to light and dark. So this is a figure of the sodium. Opsin activation changes sodium permeability to higher segment, changing the rate of the release of the inner segment synapse of bipolar cell. Changes in bipolar cell activity detected by ganglion cells located by stimulation. Neurotransmitter release bipolar cell to ganglion cell. And the ganglion cell goes to the brain for processing. So if you see brighter light, it's more destructive, less time to regenerate. Dimmer light, less destruction, longer time to regenerate. So rods take longer to regenerate. Visual issues relative to light and dark color. It does occur when sodium gates close in the presence of light, only during receiving So there is light bipolar cells, say, to send to the brain of light. Our bipolar cells inhibited and sent to the brain. The brain unscrambles it. The brain gives sensitivity and resolution. Eyeballs, retina, tells you to control light and dark. Cerebral cortex. So the cerebral cortex gives images through the thalamus and innervation 4. So it goes to the thalamus, the occipital lobe, then to association areas. Examples would be memories of a cell phone, integration areas, emotions, memory, moving to bright light from a dark room or from dark to light version. Vice versa, takes a while for you to set because rods are destroyed by light. Cones regenerate fast to see color though. That's it. I have my quiz in 10 more minutes. I got 10 minutes to shower. My roommate's playing loud music, so thank you guys for watching this video, and let's hope I do on this quiz.